Today we will start with anatomy. Okay. First, first question. All of the following statements about seminal vesicle are true except stores the spermatozoa, actively depends on the level of testosterone, secretion has abundant fructose, lined by pseudostratified columnar epithelium. Except stores the spermatozoa. So seminal vesicle actively depends on the level of testosterone. Secretion has abundant fructose, that's also true. Lined by pseudostratified columnar epithelium, that is also true. So seminal vesicles are two lobulated sacs. Two lobulated sacs situated between bladder and rectum. Bladder and rectum. So seminal vesicle has two lobulated sacs situated between bladder and rectum. Each vesicle is about 5 cm long, directed upwards laterally, and is lined by pseudostratified columnar epithelium. The lower narrow end forms the duct of the seminal vesicle that joins the ductus deferens to form the ejaculatory duct. The seminal vesicles do not form a reservoir for spermatozoa. The secretion is slightly alkaline and contains fructose and a coagulating enzyme called vesiculase. So the seminal vesicle secretion from seminal vesicle they contains fructose, number one. Number two, vesiculase, a coagulating enzyme, very important. Another thing very important is that secretion from the seminal vesicle is alkaline in nature. Okay, this is spermatogenesis, starting with spermatogonium. Then it undergoes mitosis to form two cells. One is daughter cell type B spermatogonium, daughter cell type A spermatogonium. This daughter cell type B becomes primary spermatocyte. Meiosis 1 occurs. Meiosis 1 occurs and there will be two secondary spermatocytes. And meiosis 2 occurs and there will be 4 spermatids. And at last there will be 4 sperm cells. This is in short. Okay, next question. At what stage embryonic development does an embryo normally begin to implant in the endometrium? Very important question. Blastocyst, 4th cell stage, morula, trilaminar embryo. After fertilization, the fertilized ovum begin to divide. It migrates through uterine tube, undergoes cleavage till a 16th cell stage. So the fertilized ovum begin to divide and migrates to uterine tube, and it cleavage means it divides to form 2-cell stage, 4-cell stage, 8-cell stage, 16-cell stage, and at last it will become blastocyst. And the 16-cell stage is called morula. But while this happens, it travels through the uterine tube towards the fundus of uterus in order to implant there or get implanted. Now the cells of morula differentiates into an inner cell mass which is completely surrounded by an outer layer cells. Outer layer cells further give rise to structure called trophoblast. These outer layer cells is called trophoblast. As shown here, you can see these are inner cell mass inside content. This is the membrane trophoblast. This outer surface is called zona pellucida, and the cavity is called blastocele or blastocyst cavity. Now it reaches the blastocyst stage approximately 110 cells on about day 5, and it enters uterus on about day 6. Implantation begins on the day 6 with the in situ trophoblast of the embryonic pole of the blastocyst eroding into endometrium. This eroding and get implanted. Okay, so this is how this implantation occurs. This is all about the implantation, and each and every facts is a question. 16 cell stage is a morula, but 16 cell stage is morula, but blastocyst is the one which is the stage when it gets implanted. The fertilized ovum get implanted. Blastocyst is reached. It forms on day 5. On about day 5, and it enters the uterus on day 6. Then implantation normally begins on day 6 with this in situ trophoblast of the embryonic pole of the blastocyst eroding into the endometrium. So day 6, and then erodes into the endometrium or implants into which layer? Endometrium. Not other layer. They can ask this also by giving other options as confused like myometrium or null, but endometrium will be the answer. Implants into endometrium. So this is about implantation, and each and every facts can be a question. So anyway, next. Which one of the following is not a derivative of midgut? Appendix, jejunum, ascending colon, descending colon. Now let's revise the derivatives of foregut, midgut, and hindgut. From foregut, it gives rise to first 2 sections of the duodenum: esophagus, stomach, duodenum first and second parts, liver, gallbladder, pancreas, spleen, superior portion of the pancreas. Midgut give rise to lower duodenum, jejunum, ileum, cecum, appendix, ascending colon, and the first two-thirds of the transverse colon. While hindgut, so midgut give rise to appendix, ascending colon, jejunum, ascending colon, and appendix, and ascending colon. Why these three are important? Because these three are options. That's why it's bold. And the third one, descending colon, is not present in, not written, not derived from midgut. So what are the structures that arise from hindgut? Last third of the transverse colon, descending colon, rectum, and upper part of the anal canal. So descending colon is derived from hindgut. Derived from hindgut. Which of the following is not derived from the first pharyngeal pouch? Middle ear, Eustachian tube, tympanic membrane, palatine tonsils. So its answer will be palatine tonsils. Is not derived from first pharyngeal pouch. First pharyngeal pouch is located between first and second pharyngeal arc. Palatine tonsil is the derivative of second pharyngeal pouch. And Eustachian tube, middle ear, masted antrum, and tympanic membrane derived from first pharyngeal pouch. But this palatine tonsil is derivative of second pharyngeal pouch. This is from second pharyngeal pouch. Pharyngeal pouch. And these three are derived from first pharyngeal pouch. Vessels in umbilical cord. How many? One artery, two veins, two arteries, one vein, one artery, one vein, two arteries, two vein. And the answer will be two arteries, one vein. It contains two artery and one vein. So umbilical cord is the structure that connects mother to fetus and acts as a bridge. For transfer of blood and other nutrients from the mother to baby. Now it contains two arteries and one vein. Arteries carry deoxygenated blood from fetus to mother, while vein carry oxygenated blood from mother to mother to fetus. Although it is unusual for the vein to carry oxygenated blood and for the arteries to carry deoxygenated blood, this is the situation in the umbilical cord. Another this type of exception you can get in case of pulmonary veins and arteries connecting lungs and heart. So So cord contains Wharton's jelly and the structures that are embedded. Wharton's jelly. This Wharton's jelly actually what it does, it's a first of all gelatinous substance, gel-like substance. Okay, gel-like substance, and this largely this is made up of mucopolysaccharide obviously, and protects made up of mucopolysaccharides, and it protects the blood vessels inside. Protects vessels inside. So inside of this cord, umbilical cord, there are two arteries and one vein, allantois, and remains of vitellino-intestinal duct. Okay, next question. Gallbladder is lined by ciliated columnar epithelium, brush-bordered columnar epithelium, strata columnar epithelium, pseudostratified columnar epithelium. So the answer will be brush-bordered columnar epithelium. Now, microvilli, when microvilli are of same height, arranged regularly, they are termed as striated border, as observed in small intestine. And this is the histology of the gallbladder. Submucosa, adventitia, muscular layer. This is the epithelium. Mucosal fold. These are mucosal folds, and this is lamina propria. So in fact, you must know the name of these structures. Like this is cystic duct of gallbladder, and this is common hepatic duct, and this is right hepatic, left hepatic duct, and this is common bile duct, CBD. Common bile duct. So common bile duct is formed by cystic duct and common hepatic duct. This is right hepatic, and this is left hepatic duct. Okay. And another thing is that body of the gallbladder, fundus of the gallbladder, neck of the gallbladder, cystic duct. And this is Callot strangle. This is Callot strangle. This is Callot strangle. So in the recent FMG also this happened within 3 to 4 years. In one or two or three of the FMGs, they gave these structures, and they simply mark this A, B, C. So and asked what is the name of C, structure C, common hepatic duct. So common hepatic duct or common bile duct or cystic duct or any other name they can give. So in this way, questions can come IVQ from anatomy. IVQ anatomy maybe like this also. Very simple, but you must know the name, otherwise very easily 1 mark will go. And also the histological names, structural names. Another thing, the main thing of this question, gallbladder lined by brush bordered columnar epithelium. So you see the options, all are columnar cells. All are columnar epithelium or columnar cells. But the question was ciliated, brush bordered, striated, or pseudostratified. And the answer was the answer is brush bordered. Okay, next question. Hassel's corpuscles are found in liver, spleen, kidney, thymus. And answer will be thymus. Corpuscles mean any small body. Hassel's corpuscles is spherical ovoid body. Spherical ovoid body found in thymus medulla. Medulla of the thymus, composed of concentric arrays of epithelial cells containing keratohyalin bundles of cytoplasmic filaments. So Hassel's corpuscles are what? They are spherical or ovoid bodies, and they are found in middle of the thymus. They are composed of epithelial cells that contains keratohyalin and cytoplasmic filaments. So basically, they are composed of epithelial cells containing two things. Number one, keratohyalin, 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 and number two, cytoplasmic filaments. So some other known corpuscles are laminated corpuscles, Malpighian corpuscles, Meissner corpuscles found in nephrons is Malpighian. Meissner's nerve ending in the skin, chiefly palms and soles. Then passiniform corpuscles responding to muscle stretch and light pressure. Passiniform, very important. Red corpuscles, red blood cells, and white corpuscles, white blood cells. So this each and every structure, each and every name has to be remembered. Where it is found, nephrons and spleen in white pulp. Where? In white pulp, Malpighian corpuscles. So these are all important questions. It can be framed in a question. Okay. So each and every name and its locations are very important for FMG. Yeah, cord of Billroth. Cord of Billroth is seen in liver, spleen, kidney, thymus, and the answer will be spleen. Cords of Billroth are also known as splenic cords. Also known as splenic cords or red pulp. Red pulp cords. They are found in red pulp. These red pulp cords are found in red pulp of the spleen between the sinusoids consisting of fibrils. And connective tissue cells with a large population of monocytes and macrophages. This cord contain half of the human body. Human body is monocyte. So cords are found in the red pulp. And this is the white pulp. Marginal zone. This is the marginal zone. This is the primary follicle. This is the primary follicle. Periarteriolar lymphoid sheath. Arteriolar lymphoid sheath. Germinal center. This is the vein. This is the artery. So cord of Billroth. Not passing through medial epicondyle. Nerve passing through medial epicondyle is ulnar, radial, median nerve, posterior interosseous nerve. And the answer will be ulnar nerve. See the ulnar nerve from here to medial epicondyle. Okay, and this is radial nerve near about. This is the medial epicondyle. Here ulnar nerve is passing, while radial nerve is passing through lateral epicondyle. And now the median nerve This is the median nerve. So now passing through medial epicondyle is ulnar nerve. We can see in the picture. So when we hit our elbow on any surface, the current-like feeling in arm, is due to ulnar nerve injury. Medial epicondyle fracture causes injury to ulnar nerve, obviously. Ulnar nerve palsy causes partial claw hand. Very important. Partial claw hand, sensory loss of medial elements. Okay, so claw hand. In claw hand, what happens? Sensory loss of medial, not 11, 1.5 digit. 1.5 digit, okay? Rough estimation is up to this. Half of the ring finger and little finger. So weakness of the grip, paralysis of intrinsic muscle. This will be sensory loss. Sensory loss. Loss of flexion of MCP, metacarpophalangeal, lumbricals and interossei. Loss of adduction of thumb. Okay. Okay, so this is ulnar nerve palsy. And fracture causes injury to ulnar nerve. Medial epicondyle fracture causing injury to ulnar nerve injury also causes weakness of the grip, loss of MCP flexion, lumbricals and interosseous, and loss of adduction of thumb. Loss of adduction of thumb, and this function is also lost. But sensory loss occurs only one half, little finger and half of the ring finger. Anastomosis of subscapular artery are formed by all except transverse cervical artery, suprascapular artery, first part of subclavian artery, second part of axillary artery. And answer will be second part of axillary artery. So the extreme mobility of the shoulder results in kinking of the axillary artery. Extreme mobility of the shoulder results in kinking, means pressure on the axillary artery, and to compensate this, the arterial anastomosis occurs. Exists between branches of first part of subclavian artery and third part of axillary artery, not the second part. First part of subclavian artery, anastomosis with third part of axillary artery. It provides collateral circulation when the subclavian artery is obstructed. This anastomosis involves suprascapular artery and deep branch of transverse cervical arteries. So this anastomosis involves suprascapular artery and deep branch of transverse cervical arteries as well, both arising from thyrocervical trunk of first part of subclavian artery. Now subscapular artery is circumflex scapular branch arising from third part of axillary artery. So anastomosis involves what? First of all, suprascapular artery, number one. Suprascapular artery. So all are arteries. I'm not writing arteries and arteries. Okay. Now suprascapular artery. Number two, transverse cervical artery or superficial cervical artery. Transverse cervical arteries. Both they arising from thyrocervical trunk of first part of subclavian artery. Thyrocervical trunk from first part of subclavian artery. And this anastomosis with subscapular artery, and its circumflex scapular artery and circumflex scapular scapular branch of subscapular artery. Scapular branch. And this subscapular artery arising from third part of axillary artery. So to be to say on a major structure, it's an anastomosis between first part of subclavian and third part of axillary artery. And to be very specific, first part of subclavian artery from first part of subclavian artery, suprascapular artery comes out, transverse cervical artery comes out. These two artery joins with subscapular artery. These two artery joins with circumflex scapular branch of subscapular artery, and in turn this subscapular artery came from third part of the axillary artery. So on a major on a broad division, third part of axillary artery and first part of subclavian artery anastomosis. And to be specific, it becomes suprascapular and transverse cervical with circumflex branch of subscapular. Okay, and this anastomosis occurs due to compensate compensate the normal blood to compensate the blockage of axillary artery blockage, or we can say disruption of normal flow of the axillary artery, which occurs due to the mobility of the shoulder. Extreme mobility of this shoulder may kinking. Actually, kinking is not exactly pressurized due to too much of fold. What happens? It like fold like occurs like this. So in this area, the blood flow will decrease. And to compensate all this, to compensate the blood flow, another other vessels anastomose with each other, so that the normal flow or the normal demand of the blood can be met up. Okay, so this is a very important anastomosis. Next, medial boundary of the anatomical snuff box formed by the external pollicis brevi, extensor pollicis CIS. It will be pollicis brevi, extensor carpi radialis longus, extensor pollicis longus, extensor carpi radialis brevi. And answer is pollicis longus. The medial boundary. Okay, so surface anatomy. This is the snap box, right? The boundaries are formed by tendon, basically tendon of extensor pollicis longus and extensor pollicis brevi and abductor pollicis longus. So the anatomical snap box is formed by tendons of extensor pollicis longus and brevi. Brevi. Extensor pollicis brevi. And abductor pollicis longus. Abductor pollicis longus. The posterior boundary, medial border by if the hand is, just imagine, if the hand is normal anatomical position, then this portion will be medial. This boundary will be medial boundary, and this portion, this boundary is lateral boundary. So medial boundary is formed by tendon of extensor pollicis longus, and to be specific, tendon of extensor pollicis longus. And the lateral boundary is formed by extensor pollicis brevis, sorry, extensor pollicis brevis and abductor pollicis longus. Abductor pollicis longus. Obviously, tendon. Tendons of extensor pollicis brevis and abductor pollicis longus. The proximal border is formed by styloid process of radius.